Okay, in this is the experiment from paper 5, plant physiology and biochemistry. So the aim of the experiment is preparation of standard curve for the estimation of starch by I2-KI method. Now, coming to the principle, here we are going to use the I2-KI reagent. Alright, now starch, as we know, you have already studied in your theory as well. I had explained that starch it is made up of amylose and amylopectin. Actually, what happened here, this starch it will react with this reagent to give a deep blue colored complex known as the starch iodine complex, having a maximum absorbance at 580 nanometer. Now actually it is the iodine from this I2-KI reagent, the iodine it will react with the amylose part of starch and not with amylopectin. Alright, so here what happened in solution, this potassium iodide in solution it will exist as these two ions. Then this iodine ion it will combine with iodine to give rise to a triiodide ion, and actually it is this triiodide ion that will react with the amylose of starch. Alright, so here because the iodine, this iodine it does not react directly with starch as it is not very soluble in water. So this is the reaction. As you can see, when this triiodide ion forms, it will then intercalate itself into the coil structure of the amylopectin. As you can see here in this figure, you can see this one pink in color, it is the triiodide ion, and it intercalates itself, or this iodine it will slide into the starch coil. Alright, this is the amylose starch, and you can see when the iodine inserts itself in the structure of the amylose, then it will give rise to the starch iodine complex. Alright, which is deep blue in color. So here this I already explained that iodine it does not react directly. Why? Because it is not very soluble in water. In the presence of potassium iodide, you can see that a linear triiodide ion complex is formed which is soluble in water, and it is this triiodide ion which will react with the amylose of starch to give rise to a starch iodine complex. So coming to the requirements for this experiment, the first thing that we need for this experiment is the starch solution. Then we need the I2-KI reagent, the glasswares and pipettes, and also the calorimeter, the photocalorimeter for taking the absorbance reading, right? The optical for measurement of the optical density of the starch solutions, and the glasswares and pipettes such as the test tube racks. Then we require pipettes and conical flasks, beakers, measuring cylinders, and all these things they require in this experiment. And also this is a photocalorimeter, for a digital photocalorimeter for taking the absorbance reading. And coming to the preparation of the reagents. So the first reagent, that is the starch solution. Of course, we need a digital weighing balance for measuring the chemicals. So the first reagent that we are going to prepare here is the starch working solution, and the standard concentration that we are going to use for this experiment is 50 microgram per ml. So for this experiment, for this reagent, we will take 100 milligram of starch in 50 ml. First, we will dissolve in 50 ml of distilled water. And this solution, as you can see, the starch, it does not dissolve properly. So for this, we have to boil it for 1 minute. After boiling, we will boil until we get a clear solution of starch like this. Okay, we will boil for 1 minute, and then we will make the final volume 200 ml with distilled water. Alright, we will make it the volume 200 ml with distilled water. So this will be your stock solution. Alright, starch stock solution. So for using in the experiment, we will dilute the stock solution 20 times. Okay, 20 times dilution by taking 10 ml of this stock solution with 100 ml of distilled water. So this will be our working solution for starch. And the next reagent that we require is the I2-KI reagent. So for this, we need 0.6 gram or 60 milligram of potassium iodide and 60 milligram of iodine in 10 ml of water. That will be the I2-KI stock solution. So this is the stock solution. We need to dilute this stock solution for using in the experiment. So for working solution, we will take only 1 ml of this stock iodine solution with 100 ml of distilled water. So this will be the working solution. Now coming to the different procedures. So first thing what you will do, you will keep the test tubes in test tube rack, and you will mark the test tubes according to the concentration of the starch solution that we will be using. So we are going to prepare concentration gradients of starch, different concentration gradients of starch in these different test tubes. So you will mark accordingly along with the blank and also the unknown. So the first step is that the concentration gradients of starch solution. So for this experiment, we are using this concentration gradients 5 to 50 microgram per ml of 2 ml each, were prepared in test tubes by diluting the starch concentration with distilled water. So what you will take here, you can see in the first test tube, you will take 0.2 ml of starch with 1.8 ml of water. Here you will take 0.4 ml of starch with 1.6 ml of water. So the volume will be 2 ml in all these concentration gradients in all the test tubes. And also a blank was prepared by taking only distilled water 2 ml. So the next step, we will add, after diluting with water, we will add the reagent, the I2-KI reagent, 0.5 ml to all the gradients of starch solution, including the blank and also the unknown starch solution, different concentration. Here we are taking 2 concentration of starch in 2 different test tubes, were prepared in the same manner in 2 separate test tubes, and also the reagent, I2-KI reagent at 0.5 ml will be added to the unknown test tube as well. So when you add the reagent, what happened? The reaction will takes place, and thus you will get this blue color. And as you can see that the intensity of the blue color in these different test tubes, it is different due to the different concentration of starch solution. Here, since the concentration of starch solution is less and here it is more, you can see that the blue color formation is less and here it is more intense. Alright, so you can see that the intensity of the color, it increases with the increases in starch concentration. So after you are getting this reaction, this blue colored complex, the next step is to take the optical density of all these test tubes in this using this photocalorimeter at a wavelength of 580 nanometer. After taking the optical density, we have to prepare a standard curve by plotting this optical density against different concentration gradients of the starch solution. After preparing a standard curve, the concentration of the unknown will then be calculated was estimated by plotting the optical density from the standard curve that you obtain. And this is the table. See, alright, in the first column here, you will write the different starch concentration that we are taking in different test tubes, that is from 5 to 50 ml. And since these two we do not know the concentration, so you will write as unknown 1 and unknown 2. In the second column, that is, we will write the volume of the starch working solution that we will be taking. So for the blank it will be 0, and all these concentration from 0.2 to 2 ml. And for the unknown, actually in the lab we already told you the unknown, how much to take, and the volume of distilled water. But actually in during the exam you will be provide an unknown sample solution. Alright, so you will not know what is the volume of the starch and the distilled water. So you will leave it as blank, since this is the unknown. We will find it later on, and we will provide you the unknown. You will just add this I2-KI reagent to the unknown as well. And as you can see in the fourth column, we will write the volume of the reagent, then the absorbance reading, since we are doing in replicate, so it will be R1, R2. And for calculating this formula, we will be taking the mean of all these values. Then this value here in this column, the absorbance of both, the mean value of both R1 and R2, we will be calculating using this formula. Okay, for plotting the graph here, that is the OD of the sample minus OD of the blank. Alright, so after calculating all these values, then these values we will plot in the graph to get the standard curve. Now the standard curve, okay, this is just an example. Alright, this is not the one that I plot from these values on the table, but this is just to show you a kind of similar graph you will get. Alright, similar to this one. But as you can see here from the graph, that some of the values here, they are not exactly on the standard curve. So when you join all the points, you will find that not all the points they lie on the graph. This is due to the errors while doing the experiment, the handling errors, the pipetting errors, and many errors while doing the experiment. That is why you should always be careful in order to try to minimize the errors so that you will get a good curve. So, but as you see that the errors are not so much, as all the points they lie very close to this standard curve. So after getting the standard curve, we will find the unknown by taking the absorbance reading of the unknown. Suppose if the absorbance reading of the unknown is here, we will draw a perpendicular line from the y-axis to the x-axis, and when it touches the standard curve, we will drop a perpendicular line to the x-axis. So this value here, it will be the value, the volume of the unknown that you are taking. Since you will not know, we will provide an unknown sample solution to you, unknown starch solution. So you will first know the volume of the unknown starch from the x-axis, from this graph. Then that value you will calculate the concentration from this value here. For example, suppose if it's 1.4 something here, this value you will multiply with the standard starch concentration, that is 50 microgram per ml. You will get the concentration of the starch. So after getting the value, you can see that the starch concentration, for example, we know that the optical density of the unknown one. Here I'm just showing you an example for the unknown one, how to calculate. You see it is 0.13, which corresponds to, for example, it's 1.4 ml. Alright, just an example, if you get this value from the x-axis 1.4 ml, then this value here you will multiply with 50, which is the standard concentration of starch, and you will get the value of the concentration of the unknown. Alright, so this is how you will calculate the concentration of the unknown starch solution. Now coming to the discussion, this we had already discussed that the iodine, this triiodide ions, it will react with the amylose. Alright, it will react with the amylose of starch and not the amylopectin. It will react with amylose, as you can see in this figure, that this iodine complex, pink in color, it will insert itself in the middle, in the center of the amylose coil. Alright, to give this blue color complex, that is starch iodine complex. And however, the reaction does is not a chemical reaction, it is a physical reaction where this triiodide, it intercalates in the coils, within the coils of this amylose. And also starch, as you have learned from the theory, that it generally contains 20 to 25% amylose and 70 to 80% amylopectin. So in different test tubes, if you remember, we are taking a small volume of starch, for example 0.2, then 0.4, and so on till 2 ml. So as the concentration of starch is more, so also the concentration of amylose is more and more according to this concentration of starch. So the intensity of the color also, or the strength of the blue color, it depends on the concentration of the amylose present in that starch solution. Alright, so that means the intensity of the color, alright, the intensity of the blue color or the strength of the blue color, it increases with increase in starch concentration. That means in amylose concentration, as starch is made up of amylose and amylopectin.